So we have defined evolution as a change in allele frequencies over time. So what's going to cause allele frequencies to change over time? Well, one thing is mutation. Another thing is non-random mating, meaning organisms tend to breed with those usually nearby themselves, okay? Rather than those further away, things like that. Selection is going to cause evolution, and this could be natural selection or a subcategory of natural selection that we call sexual selection. Genetic drift causes evolution, and so does gene flow. We're going to define all of these shortly. But what causes evolution? All of these things. So if you want to stay in Hardy-Weinberg equilibrium, you can't have mutations. You have to have only random mating. You can't have selection. You can't have drift, and you can't have gene flow. Okay? Some mutations we've already introduced. That's where new alleles arise. Genes get copied. Genes get rearranged. There are point mutations, things like that that change the phenotypes of organisms. Gene flow is when genes flow from one population to another. Think of it as the effect of migration. So here we have population A and we have population B. They have different proportions of phenotypes. Here it's 20 blue to 10 red, and here it's 10 blue to 20 red. And so some of these organisms randomly leave this population and flow to this one, bringing their alleles with them. And some of these leave this population and go to this one. So they're going to change the genetic makeup of the populations that they're migrating to. That's called gene flow. I like to think of it like a river and a boat on a river. Here's the boat. It's going from point A to point B. It's moving from point A to point B, right? That's how I tell flow apart from genetic drift. Genetic drift is like a boat that's randomly going back and forth on the ocean, not going anywhere. Drift, by definition, is random changes. We're not talking about natural selection here. We're talking about random changes. And we're going to focus on two kinds of drift. One is called the bottleneck effect. That's when you have maybe a red organism, a couple of red organisms here, and maybe you have a couple of blue ones, and you have a purple one. Okay? Here's your population. Then a big wildfire comes through and it kills everybody, but the purple one happens to survive. That means the next generation is all purple. That's called the bottleneck effect. It's like you funneled down and you only let some of them survive. Like in this wild boar example, there's lots of diversity here, but only little b survives after something happened, maybe climate change or something. Same thing can be said in real life for a lot of wildlife species that have almost gone extinct and then recovered, like some of our big cats. We have a loss of diversity. Here's an example from your book showing a non-biological example. We have white balls, blue balls, and yellow balls. And if you only shake a few of them out into a surviving, a bottlenecking event, the population that survives is still mostly blue, but I think there's fewer white than you might have expected from this original population, and there's no yellow. So you only have some of the original population surviving, and their allele frequencies are different than that original population. Another example of genetic drift is the founder effect. That's when you have a population here, population A, and you have an island or something over here, or a new continent, and a few random individuals from population A move to this new place and start a new population. But their allele frequencies of this random sample that moved are a little bit different than the population that gave rise to them. So here, for example, in the original population A, we have 20 pink to 10 red. That's 20 to 10. But in the new population, it's a 1 to 3 ratio because it was random. These three happened to fall on a log and wash away to a new island and start a new population. They are the founders of the new population, so there's a new set of allele frequencies here. We can see this in some Amish communities in Pennsylvania, where some members have an extra finger on their hand because one of the original founders of that community was someone who happened to have an extra finger on their hand, and they are the ancestor of almost everyone in that population. So therefore, that in that population, you have folks with an extra finger. The starlings that we have here in North America were introduced from only I forget, something like 10 birds from England. England has millions of these things and lots of diversity, but someone just randomly grabbed 10 of them and let them loose in New York 100 years ago or more. And so every living one in America now is a descendant of those 10. They don't have nearly the genetic diversity of the original population back in England. That's the founder effect. Random mating. That's when every member of a population has an equal chance of mating with another. So this will actually stop evolution. Well, it's one way to prevent evolution. Because if this individual, let's say this individual has an equal chance of mating with this one or this one way over here, right? That's what we're saying. Random mating. It's equal. They randomly breed with everybody. The important thing to note here is a lot of times organisms that are close to one another physically are related. And over here, you might have a different family. So if you mostly breed with those close to you, you're going to have a lot of inbreeding. And if you don't have random mating, you tend to have inbreeding, which is going to change your expected phenotype frequencies. Why? Well, you tend to have similar genes to those that are related to you. So for example, you could be big R, little r, whereas the whole population has big R, little r, and little r. You are going to be more closely aligned to those near you, and you're not going to create as much diversity when you have offspring if you're closely related. And while we're on this subject, let's introduce why inbreeding is not good for you, for a population, because more of your offspring are going to get sick and die probably, which will also change the phenotype situation. Why is it likely that more will get sick and die? Well, a lot of diseases are only show up when you have two copies of the recessive allele. So let's say you are little r, and your cousin is little r, and you have offspring together. A quarter of your offspring are going to be, I'm sorry, I think I said that wrong. Let's say you're big R, little r, your cousin's big R, little r. You have offspring. A quarter of your offspring will be little r. That's an increase in homozygosity. And if little r, so if a homozygous recessive condition causes a disease, you're going to have a lot of that disease in your offspring. I have an example here of a woman that looks blue because there was a condition in a family in Kentucky where there was some inbreeding going on in this one part where people didn't get in or out much, and they had hemoglobin that had the wrong shape. Their O2 receptors were not very good. They couldn't carry oxygen very well on their hemoglobin, and they looked blue. And you can even see that in old pictures of the family. You can look them up, the Fugit family. This is a thing that affected the family because of inbreeding. Now, if this had been a deadly disease, everyone who you see there in blue would have not survived childhood, right? So let's go back to this real quick. We're going to spend a little more time with selection shortly, but mutations, they cause evolution. Non-random mating causes evolution. That means you're mating with those nearby. Selection causes evolution. That means, for example, some of you are better at surviving and having babies than others. That causes evolution. Drift and flow cause evolution. You should be able to define each of those things in your own words, okay? In the next video, we're going to focus on selection.